Okay.
And so she doesn't have to introduce herself.
No.
Too late. Our next speaker is Dr. Teresa Casey on the role of clocks in memory development and lactation.
So just to be clear, I didn't invite myself to speak. I'm really part of the endocrinology meeting, but we merged them both. And I'm really happy to talk about something that I've developed a huge passion for, the beauty of life and evolution and being integrated with our Earth. And I hope you appreciate the system as much as I do, or maybe even 1% of what I do. And then we'll go on from there because I'm highly enthusiastic about this system. I try not to bore you too much. So my journey into the system started with transcriptome analysis. And we had wondered if there were other systems other than hormones that may be regulating these homeoretic processes. And as we've talked about homeoresis, I'm lecturing to the choir right now, but it's the coordinated changes in dam physiology as she to support the dominant energetic needs of lactation, but it's also involved in growth and photoperiod responses and even heat stress response. So, but we were asking in particular what else was maybe regulating these changes in the dam. And so what we did is we decided to take, I don't even know. We had said, okay, there has to be something other than hormones. And so we decided to take an untargeted approach and use transcriptomics. And our hypothesis was that if there was another hierarchical system, that system would change across multiple tissues as the dam transitions from late pregnancy to the onset of lactation. And what we did at the time, we were using rat dams, and they were late pregnancy. So pregnancy day 20, rats have about a 23-day gestation, and lactation day 1, and we isolated RNA from mammary, liver, and adipose tissues and looked for genes that were commonly changed as the dam transitioned to lactation. And of course, you know the end of my story. What we found is circadian clocks genes changed commonly in the same direction as the dam transitioned. And so then that's okay. So I'm done with my story. They regulate homeoresis and also regulate homeostasis, and that's the end. But I'm going to take you on my journey of 12 years of research, sorry, and tell you just a little bit about circadian clocks. Circadian clocks basically regulate everything. They underline everything in our life. They're highly integrated with our metabolic systems, our reproductive systems, and they are they reciprocally regulate each other. The circadian clocks generate our 24-hour rhythms of behavior and physiology, and it does that to integrate our internal physiology. So, right, that things occur at the right time in relation to each other, and also in relation to our outside world. And that's really important. And remember, one of the worst things for us is the light that we have available to us all 24 hours a day. It's actually not good. So stop looking at your phone in the middle of the night. Actually, I told my husband I'm not going to diverge, so just keep going. Anyway, I like to view clocks as having 3 components. This is not coined by me, but it helps me simplify and to think about these things as clocks have an input. So what's the input? Well, it's temporal information, information about time, light, our food availability, exercise, social interactions, sleep. These all come in together and they say what time it is. Then we have a molecular clock that integrates this temporal information, and it results in output. And our output are circadian rhythms of gene expression that are translated into circadian rhythms of proteins that are then do chemical reactions. And so circadian rhythms of our metabolism and metabolome. In mammals, the clocks are regulated hierarchically. And what I mean by this is we have a clock in every cell of our body and every organ of our body, but there's one clock that rules them all, right? And that clock is located in our brain, in the hypothalamus of our brain. And we have suprachiasmatic nuclei. So, you have the paraventricular nuclei. Well, you also have the suprachiasmatic nuclei. And these clocks, the primary input of them is the light-dark cycle that comes from the rotation of the Earth. Again, we also have body mass is a really, in energy balance is a really important input to that, timing of our food availability and social stresses. It takes all that temporal information, integrates and says, hey, this is what time it is, body. And the way it communicates to the other cells is it sends out circadian rhythms of things like cortisol, melatonin, and core body temperature is a really important primary output of our clock. And it goes, the master clock, and it goes and says, hey guys, this is what time it is. Those in the clocks in the peripheral cells, like the mammary gland and the liver, take that information, integrate it, as well as other information may be important for its function, and then generate circadian rhythms of gene expression down the line. Anywhere from 5 to 10% of genes expressed in a tissue show circadian rhythms, and they are related to organ function and specific to that organ's function. So what is the clock? What's the missing black box? Well, our circadian timing system is a homeostatic system. So like all homeostatic systems, it's composed of some sort of feedback loop that turns on and then off. So this feedback loop is a transcription translation feedback loop. So we have positive regulators, and those are BMAL1, sometimes called arnitol, that heterodimerizes with clock or clock's paralog, which is NPAS2, functions as a transcription factor, binds to enhancer box sequences in clock controlled genes, and drives transcription of 5 to 10% of the genes, including the transcription of their own negative regulators, the period and cryptochrome genes that are translated into proteins that heterodimerize and block clock and BMAL1's transcriptional activity, resulting in circadian rhythms of gene expression. So as I said, we had hypothesized that the circadian timing system regulates the physiological and memory specific changes that occur during the transition from pregnancy to lactation. And so being a reductionist biologist, I said, okay, well, if that's true, if I get rid of the system, it has to affect lactation competence. And simply what I mean by lactation competence is the ability of the dam to support the growth of her single neonate, her litter, or in the case of dairy animals, is her milk production capacity. And so I turned to mice for some of my first studies, and what we use is this clock delta 19 mouse, and the delta 19 mutation here, it prevents it from transactivating transcription with BMAL1, so the targets are downregulated. This mouse also shows loss of behavioral rhythmicity. They also have altered glucose homeostasis, and I say this, they develop hypoglycemia or hyperglycemia and resistance. And some of this probably has to do with the diet they're on. And I bring this up because my cows behaved differentially. Of course, nobody behaves great, but they have a propensity to develop obesity on a high-fat diet or not. And anyway, so there was a lot of work done on this mouse and looking at their reproductive competence. So they do get pregnant. They have, normally mice have about a 3 to 4 day estrus cycle. These guys have a 5 to 6 days, but they become pregnant. They're actually seem to grow a fine litter during pregnancy, but then the trouble happens in the postnatal period, and their litters die, and they have a slower growth. And so the people looking at this were all like, they forget that lactation is the continuum of reproduction in mammals. And so they all ended there and they're like, okay, well, we don't know what's going on. And thank goodness, because I needed a job. And I said, okay, well, the maternal behavior was altered, I should say this, but it was altered in a manner that is indicative of a lactation problem because they nursed more often and they had longer bouts of nursing. And so that says, well, I'm not making enough milk for my babies. And then the other thing is they had a loss of rhythmicity in prolactin, but it shows chronic, the same levels across the day, which may be an issue, and I'm not sure, but something to look into. So anyway, as a mammary gland biologist, I said, I bred them, they all died, couldn't do anything, but I said, oh, I'll just back up, look at what's going on during gestation, because we know one of the most important thing that's happening in gestation is that mammary gland is growing, right? So we prepared hematoxylin and eosine stained tissues, mammary tissue sections from wild type and clock mutant mice. And those of you here, look here, although I can't see it now, my eyes have gotten old, but you see nice big lumens, and you might even see some fat droplets in here. And compared here, you see like really small lumens. And those of you who aren't mammary gland biologists, this is good and this is bad. So we have really great differentiation, and then we quantified it, and indeed these clock mutant mice had poor mammary development, which would explain potentially part of the problem with their lactation competence. But I realize you guys are not here about mice, but we can take a reductionist approach in cows too. And that's the nicest thing about this system. We can knock it out. We can knock out its function. And the reason is that inputs to clocks, when they're applied at inappropriate times, can disrupt the circadian timing system. And they're referred to as chronodisruptors. Most commonly, what's used is we disrupt the light, but you actually can inappropriately feed animals. You actually can make animals really fat, and that will disrupt their circadian timing system as well. So knowing this and borrowing things from studies on humans and rodents, we said, well, can I use these systems in cows? And so this was my first experiment in cows. And so for this study, we used a crossover design. We moved our cows into the Tystall facility, and we exposed them to 2 different periods. The first period was 16 hours of light, 8 hours of dark, which is the normal light-dark cycle on our farm. And in the second period, we basically exposed them to the worst shift work cycle you could ever imagine. So they were sleeping during the day or night. So every hour we changed their light dark cycle. And I know you guys are thinking, can you really mess up someone's physiology like that? And the answer is yes. So go to bed at the same time every night and give yourself rest, because it's not good for you. But anyway, I don't know if you can see, we had a small reduction in milk production. We also dramatically altered gene expression, lower de novo genes that regulate de novo fatty acid synthesis, as well as multiple clock genes. So armed with this preliminary information, some kind people on some panel about 4 years ago said, okay, we want to see what happens. What do clocks do in regulating the homeoretic response? Because I'm a mammary gland biologist and I'm really interested in mammary development, we backed up. We went away from lactation, and we decided to look at the effect of circadian clock disruption, or knocking out circadian clocks during the dry period or pre-parturition on metabolic balance, endocrine milieu, mammary development, and milk production. And then rather than using like a rotating shift work schedule, we decided to use a chronic jet lag. So control cows were also at the 16 hours of light, 8 hours of dark, but our phase shifted cows had the same amount of light. So they had 16 hours of light, 8 hours of dark, but every 3 days we pushed forward the light dark cycle. And why we did it every 3 days is work in humans and mice shows it takes about 5 days with a 6-hour shift in our light dark cycle. So those of you who flew from Europe, you have to stay here 5 days to feel okay. That's why we did it every 3 days, because we didn't want them to reorient themselves to their environment. We had hypothesized that since clocks play a central role in regulation of metabolic homeostasis, that disruption of the system would cause metabolic imbalance and decrease milk production. And we conducted 2 animal studies in successive years on the dry period. And the first was really focusing on what was the effect on the circadian rhythms of hormones and metabolites in these animals, and then the effect of the circadian disruption just during the dry period or second half of the dry period on milk production to 60 days. And in the second study, we did some pretty intensive sampling in terms of multiple biopsies and intravenous glucose tolerance test, and then looked at mammary development in those biopsies, as well as did some transcriptome analysis and milk production to 60 days. The first study had 32 cows, 16 controls, 16 phase shifted, and that's what PS stands for, or circadian disruption. And that study was headed up by the guy who's the postdoc in my lab now. He's now at Berry College, and his first months of being faculty, wonderful person, highest EQ of any person I ever met. And then the second study was headed up by a master's degree student who was in Jackie Bowerman's lab, and Jackie was really instrumental to helping me get this going, and that had 16 cows. And Conor McCabe is now at UC Davis. So for this study, all the cows were dried off at 60 days prepartum. And because I'm a mammary gland biologist, I was like, oh, I got to pick which phase I want to disrupt. So I ignored involution, right? So, and I started the study at 5 weeks before parturition. And shout out to Lorraine Sardillo, because she really helped define these stages in terms of her work. And so 5 weeks before expected calving, that's when the gland transitions to mammogenesis or regrowth. And it also, and so they had the phase shifted treatment from 35 days to parturition. And then that's just when lactogenesis. And then we did a bunch of stuff in the postpartum period, but I'm not going to show it because I have to pick things to show. The cows were both in the same barn, and they were separated by double draping of fire retardant tarps, and they had basically identical environments except for light dark cycle. They were exercised the same time of day and fed the same time of day. In the first study, we wanted to capture circadian rhythms, so we blood sampled the cows every 4 hours over 48 hours by jugular catheters, and we did that at 3 weeks before expected calving and 1 week before expected calving, or 2 weeks after initiation of the phase shift treatment, or 4 weeks. And then we also wanted to look at core body temperature, and we placed data loggers intravaginally on blank cedars and had the body temperature recorded every 30 minutes at the same time period. In the second study, exact same design and handling of the cows, but we did mammary biopsies, liver biopsies, and also muscle biopsies, but I'm not presenting on that, so I didn't show it. And we did that at 3 weeks before expected calving, and then wanted to look at insulin and glucose tolerance. And so we did an intravenous glucose tolerance test at 2 weeks. And we actually, before expected calving, and also did these things in the postpartum, but I'm not showing that. So the first thing we need to show is that our treatment had an effect. Melatonin, as you know, is highly sensitive to the light dark cycle, is also a primary output of the master clock, because actually what happens is it feeds the pineal gland, it feeds back onto the master clock. And I know these circadian rhythms are really hard to look at if you're not used to them, so I'm just going to tell you what happens here. And Karen was like, I don't like it. It goes like this all the time. So basically what I wanted when we started working in this area. But anyway, so the control cows on the top and the phase shifter on the back. But what you can see in the control cows, we have this really nice circadian rhythm. But after 3 weeks and 1 week, we have this attenuation, right? So we have this dampening in our circadian rhythms. And this amplitude is actually indicative of the underlying strength of the clock and the system altogether. So you can see our treatment dampened the circadian rhythms here. The other primary output of the master clock is the core body temperature. And the bottom line in both the graphs here and here are, that's 3 weeks before expected calving, and the top is 1 week before expected calving in both. And what you can see is the control cows have nice circadian rhythms at both time points, that you see some circadian rhythms, because you know the body really wants to maintain circadian alignment across all the systems, but we do lose it significantly in the phase shifted cows, particularly at the 1 week before expected calving. And one thing you're going to notice is both cows have an increase in basal body temperature, but only the control cows show this really big increase in amplitude. And I was like, hey, is that master clock trying to synchronize our cows in some way? And so then I am only going to show you the data on the control cows and in their hormones right now. So we said, okay, well, if it is trying to synchronize them, what's happening at the level of the hormones? So anybody who's studied cortisol, and I should have read more deeply into cortisol before I started this, is cows don't actually show a nice circadian rhythm of cortisol, and it's something to do with vigilance of rumination. But if you look at cortisol here, there's two different analyses. This is second harmonic analysis, and this is first harmonic analysis. I had to work with a mathematician. I'm not good at math. And so she helped me with all of this. But you can see we get a really good, beautiful circadian rhythm in cortisol. And nobody can pick this up, but you can 1 week before expected calving. And then this is serotonin, and our friend serotonin also shows a really nice circadian rhythm. So I was like, oh my gosh, they must be trying to synchronize the timing of parturition. And so we looked, and the cows that had the disrupted rhythms, the phase shifted cows, had prolonged gestations, about 3 days. Go back to the literature, people who look at the effect of circadian disruption during pregnancy in sheep also ended up with prolonged gestations. And there's work in rodents that say that it possibly has a pretty good role in probably timing parturition. So right there, we ignore our dry cows, we move them around, we give them a lot of stress, and lots of farmers leave those lights on all the time. That will disrupt your circadian system. So right there, that's a reason to pay attention to these kinds of things as you go forward and give advice to producers. Okay, so this is the part I will always struggle for. But if you look, I said we measured milk production to 60 days in milk. And so the orange on the top here, the phase shifted cows made more milk in the first study, but less milk in the second study. So that's stinky. I would never, based on all my available data, I only believe the second study. No, I'm only kidding you. I don't think it could possibly be circadian disruption. Maybe it did help them milk production in some way, but I don't think circadian disruption could ever be good for a cow. I'm going to argue that I like the second one because of the scientific reasons. I don't think it's good for milk production. And so I'll go on from there. But metabolically, we had a little bit of a different response. We also sampled differently because we thought we were going to have more milk production in the second study too, and I was trying to figure out why. But here, the control cows are black and the phase shifted cows are gray. And what you see is actually they develop, this is glucose, I should tell you, they develop hypoglycemia from the treatment, and that it's carried over even after the treatment is, we cease the treatment. And we literally sampled these cows every day at 6 a.m. Beta hydroxybutyrate levels were they were not significantly higher. They were maybe a little bit higher, but not significant. So it didn't really affect that. So they ended up with hypoglycemia. The insulin levels were the same between both treatments. So I don't know. Anyway, this is the one I like. And the reason why I like it is because this is this is the response I expected. And because when humans have circadian disruption, they develop insulin insensitivity or insulin resistance, and same thing in mice. And so so what you can see is when we did glucose tolerance tests, they both had the same, we'll pretend this is glucose response, and the glucose response was the same in both of them. So they both brought it down at the same rate. However, when we look at insulin, you'll see the phase shifted cows needed a lot more insulin to bring the glucose to the same spot. This is a very common response to circadian disruption. So I like it better, but I can't account for differences, just tell you I got them. And so anyway, we also looked at mammary development. So keep in mind, this is only after 2 weeks of treatment. So I just turned on and off these lights on cows for 2 weeks. And if you look here, these phase shifted cows have way less epithelial proliferation. This was measured with the index of Ki-67. Kelsey actually did all this work. And then similar to the mouse, we found lower alveolar to lumen to alveolar ratio in them. So we impaired mammary development with these treatments. So then we also wanted to have a little bit better idea what was happening at the organ level. And so we isolated RNA from the liver and the mammary gland and did paired end sequencing and used DSeq2 for our differential expression. And so I'm going to say across 14,000, 15,000 protein coding genes, our effect was highly subtle. Okay, so again, it's 2 weeks later. It was pretty subtle because if you tried false discovery rate, we're right about here. So I have done a lot of this work, and I know you need a lot of genes to make a story. So I relaxed my stringency down to 0.05 nominal p-value. And at that stage, we got about 862 differentially expressed in the mammary gland and 834 differentially expressed in the liver. I said, okay, that's good for me to go forward. I'm not going to bore you with a lot of tables. I'm going to give you my last picture. This paper was accepted 2 days ago, and so I'm all excited about it. But so red is indicative of genes, upregulated genes that were enriched in the liver by phase shifted treatment. So they were upregulated during circadian disruption. And what you want to look at here is this little relationship here is we have less cholesterol efflux and more cholesterol biosynthesis. We also have a wound healing response, and that's not good in the liver. And our beta-adrenergic receptor 2 is upregulated, and we have ER stress. So overall, circadian disruption increased hepatic expression of cholesterol biosynthesis, wound response, and endoplasmic reticulin stress genes. And if those of you who know about monogastrics and humans, that a decrease in efflux and with an increase of cholesterol synthesis, that's related to development of fatty livers in human beings. So the question is, does that increase fatty liver disease in cows? And Juan Lohr had a paper that says he thinks so too. So I deferred to him. If you want to argue that, so because it is a ruminant. And in the mammary gland, all of us mammary gland lovers, so we have the downregulated genes by phase shifted treatment enriched our lipid transport and lipid transport and metabolism was downregulated. Nearly every gene involved in that, oxidative phosphorylation, it was like crazy numbers in there, were downregulated. Remodeling, and remember, this is a really important time of redevelopment of the mammary gland. So remodeling of the ECM, lysosomal enzymes, all those fans of Christina Watson, we know this is really important during this time, and immune response is down. So the overall signature of the mammary gland is that genes that regulate tissue remodeling, lipid transport, and oxidative phosphorylation were downregulated, and at least in this study may explain our decreased milk production in addition to our poor mammary development. So I'm not going to spend a lot of time on this part because none of this work was done in production animals. It was all done in rodents and cells, and so it really needs to be investigated more in a live system, if you can do that. So I started and told you that there's 3 components. So what are the inputs? Because this is really important for us to understand, and what are the outputs of the mammary clock? So a lot of our other work has focused on this. This work here that I'm showing came out in 2014, so it's really older at this point. But what we did is we took late pregnant mice and early lactation mice. So these were pregnancy day 15, 16, and lactation day 1, which I define as, oh no, these were lactation day. They could have been lactation day 1, which is 18 to 36 hours after birth. And I have a hard time. Lactation day 1 is really hard to define in animals sometimes. But anyway, we have in pregnancy, we'll say we just have circadian rhythms of the core clock genes. So there's like 6 of them, right? So we look at them and they have this nice circadian rhythm. And then we looked at the circadian clock genes, and we see an increase in amplitude in our SCN as well in our liver. And we're like, hey, that's great. And I hypothesized that the same thing was going to happen in the mammary gland, because I was like, wow, it's putting out way more. So we should have this more robust clock. But no, that's not true. What happens? Really fun things happen with the protein level, but I'm not going to talk about it because that's not an input. But I will say we did a couple of different experiments, and we put pups on at different times and looked at prolactin levels. And this pattern of the decreased amplitude said to me, hey, I think that the suckling behavior of the neonates is influencing this clock and causing this decrease in amplitude. And instead of having nice circadian rhythms of my favorite gene, this BMAL1, we actually had these ultradian rhythms. So we had 2 peaks within that 24-hour period. And so I turned to my favorite cell line out there, HC11 cells. They behave so well. They are a lovely little cell. HC11 cells are a mouse mammary epithelial cell line. That's a normal epithelial cell line. And we transfected the luciferase reporter gene hooked up to the BMAL promoter. And the blue line is, I should say, so my hypothesis, I didn't tell you guys my hypothesis. So my hypothesis was suckling was doing this, and all of us know that suckling induces a neuroendocrine response that results in the release of lactogenic hormones. Glucocorticoids or prolactin are 2 of them. And in our system, dexamethasone, there's a fly over here. Dexamethasone is our glucocorticoid, and then there's prolactin. And so the blue line here is prior to addition to dexamethasone, but there's an arrow here. And then the red line, you can see there's a nice increase in amplitude. And the same thing happened to prolactin, but there was no effect to insulin. And so this, we weren't surprised at all about dexamethasone. Glucocorticoids are a very well-known, well-characterized input to clocks, all sorts of clocks over the body. So we expected that increase in amplitude. But prolactin, we're like, hey, that's really cool. That prolactin is an input to the clocks. And not only is it an input to the clocks, but you can, I have to say, circadian rhythm biologists had to mentor me through all this. But if you apply this prolactin at a lot of different times, you get this shift in phase. So that what we were seeing is that the mammary clock is responsive to the neonate, and that's really important because not only does it respond to the person's energetic needs, but it's responding to the energetic needs of their offspring. And so that's really cool data. That's great. And so then the other thing we did, well, I didn't do this work at all. I have to promote Kevin Haring because we're working in parallel, and he's done some really beautiful work. So if you like circadian rhythms and circadian clocks, he's done some really nice work. So his work was kind of launched off of observations made in rodents that the timing of food availability functions as a powerful input to liver clocks. And in fact, it's such a powerful input, even though we all like to be ruled by the master clock, you can go rogue if you feed food to mice only available during the daytime, and you can completely flip the timing of the liver clock opposite of what the whole rest of the clocks should be in your body. And so it's not a great flipping, by the way. So night eating syndrome is a big thing in human beings, and this is not good for your circadian timing system. But he then said, okay, well, does the same thing happen in the mammary gland? And so he performed these studies with rodents and mice and found, yes, the mammary gland can well, I don't know if he said it can disassociate because he didn't look at this guy here, but or this girl here, because they're all girls. But he looked at the mammary clock in the cow, and he found if you restricted feeding to different times a day, you could flip the clock. And then he showed that when you're flipping the clock, you flip the time of de novo fatty acid synthesis in the gland. And I would support his de novo fatty acid synthesis because we see that as a probably good clock target. Okay, very busy, I know. And I'm already like saying to myself, why did I put that in? But I could draw it with IPA, and I did. But we did this other study, and the study like almost killed me, but we looked at the clock targets by doing ChIP-seq analysis for the BMAL1 transcription factor. And I only did it in cells. And I say to myself, I'm going to do that in live animal tissue sometimes, but I don't think that's a good idea. But anyway, and I say I didn't do any of this work, but my students did it, and they were amazing. And you can't see anything in here, but I do want to point out something to you. And this is really cool. So the work was done in the mammary epithelial cells, and what you see that's identified as a target is right here, prolactin, and HC11, our epithelial cells make them. Laura will tell you the mammary glands and endocrine hormone, endocrine gland. So we make our own prolactin receptor, and JAK-STAT. They're all targets. We're like saying, okay, we're going to coordinate the expression timing of genes that are important for milk synthesis, along with a bunch of solute transporters and ABC transporters. So the temporal synchronization of hormonal signaling and nutrient transport for milk synthesis is all done by that clock. That clock is pretty amazing. And way too busy of a slide to explain to you, but I will say the temporal separation is really important of incompatible processes. And in fact, this is why they think the clock originally evolved, because oxygen is toxic to our cells, right? And so you can't have high levels of oxygen happening at the same time of DNA synthesis, because we would hurt our DNA, right? And before we had eukaryotes, we had prokaryotes with no cellular organelles for spatial separation. So the only way to separate incompatible processes was by time. So that's how they think it evolved. It's really cool. Go to a circadian rhythm biology meeting. You learn so much, and they're cool people too. I was like, oh, maybe I'll switch. I like the mammary gland though. And anyway, so right now we're at a pretty elementary stage of our understanding of clocks and the regulation of the mammary gland. Those of you who have children or don't and have been to their concerts, you know they're not temporally synchronized at all. But this is where I actually think if we keep studying the beauty of like things coming in together and keeping them apart, it's like a symphony, right? And yeah, see, I'm going to start to cry about my stuff. But anyway, I think we can maximize efficiency. I'm just even paying attention to what we evolved with, right? Optimize it, cow welfare, and potentially the biggie one, decrease our impact on the environment of our production systems. So if anybody wants to study it, I encourage you. Kevin and I are all alone right now. And it's actually a great system. I have lots and lots and lots of people to thank because they did all the work and I come up with bad ideas. But so anyway, if anyone has questions, I would like to answer them.